I'm going to be talking about the process of meiosis. Most sexually reproductive animals are diploids, which means that they have two copies of every chromosome, one of maternal region and one of paternal region. And this is needed to make gametes that are haploid, which means they have one copy of each chromosome, and these gametes are needed to reproduce. And basically, two haploid gametes, which is usually an egg plus a sperm, and then plus fertilization equals a diploid zygote. And another term is homologous chromosomes. Homologous chromosomes basically have the same structural features like size, banding pattern, centromere position, and they also have same gene at the same loci positions, which means that the genes are the same, but then alleles may have some differences. And so now actually talk about the meiosis process. Meiosis is basically the process of gametes or sex cells being made in reproductive organs, and it consists of two cell divisions. The first one being a reduction division of diploid nucleus to form a haploid nuclei, and the second process is when they separate the sister chromosomes. It's preceded by interphase, which is basically when the replication of DNA is to create the chromosomes, which forms the genetically identical sister chromatids, which will be used in meiosis. So the first part of the process, meiosis 1, is when the homologous chromosomes pair up. Prophase 1 is when the homologous chromosomes pair up to form a bivalent tetrad, and they're held together at the chiasmata. And crossing over of genetic material between non-sister chromatids also happen here, like new genes, to form a new gene, which is recombination. That's basically the picture I'm going to use over a few times. And the second and third circle show like the connecting and the recombination. And continuing of meiosis 1, metaphase 1 is when the homologous pairs line up along the equator of the cell. Anaphase 1 is when these chromosomes split apart and move to opposite poles. And telophase is when cells split into two haploid daughter cells as cytokinesis happens at the same time. Meiosis 2 is actually very similar, but instead of homologous chromosomes, the sister chromatids divide into separate cells. Prophase 2, the spindle fibers reform and reconnect to the chromosomes. And metaphase 2, the chromosomes line up and along the equator of the cell. Anaphase 2, sister chromatids split apart and move to the opposite poles. Telophase 2, cell splits into two as cytokinesis at the same time. And so the sister chromatids may no longer be identical because of recombination, which makes them unique. And so there's 4 genetically distinct haploid daughter cells, which gives like the genetic kind of difference between each haploid cell, which is the bubble where the two X's are separate and they further into split into like one each. And the way I remember this is basically remembering prophase, metaphase, anaphase, telophase, PMAT. They pair up, line up, split up, and split up again. So it's just a way to remember, but it's not very accurate. It's just like a simplification of what happens, which is basically pair up, line up, split apart again. And yeah.